on the OTCQB. Thanks very much also to the conference organizers, our friends at Noble, for inviting us to come back and talk to you about ProMIS. I'll be making some forward-looking statements. I see some familiar faces, so I'll start with a bit of an overview of the company, but then I'll drill a little bit deeper into a few areas that I think are a very important part of the story. We're a company creating a portfolio, a broadening portfolio of monoclonal antibodies focused on neurodegenerative and other misfolded protein diseases. And what we bring to the table is a unique capability of creating antibodies that are highly selective only for the misfolded form. Your primary theme of what I'll go through in the next 25 minutes is that that's critical for best-in-class therapy, that's critical for value creation, and ProMIS has a unique capability there. We first applied that capability in the Alzheimer's field, creating an antibody that's scientifically superior to Biogen's aducanumab. There's been a lot of confusion about Biogen's aducanumab. I'll help to clear that up, hopefully. But we've created a version that hopefully will be better than Biogen's aducanumab, which we think will be the first approved disease-modifying therapy in Alzheimer's. Once we had accomplished that as a company, we started diversifying our portfolio, looking at other targets in neurodegenerative disease that are of great interest to large pharma. We have a number of partnering discussions ongoing in those categories: alpha-synuclein in Parkinson's, TDP-43 in ALS, and tau in Alzheimer's. I'll also talk in a moment about a couple of trends in the field that have changed the economics and the degree that one can move rapidly and cost-effectively to proof of concept and approval. That's gene therapy and the use of biomarkers. All of the above gives us a number of catalysts over the coming months and years, and I'll end the presentation with a quick overview of what some of those catalysts might be. But let me start, before I dive into more depth about ProMIS, with a little bit of a foundation of the neurodegenerative disease space. Start with that quote in the lower right corner. We agree, obviously. CEO of Roche said, "Neuroscience has the potential to be in this decade what immuno-oncology was in the last decade." Why do we agree with that? Why is there a good case for that? If you look at the items on the left in green, there's been a tremendous advance in the understanding of the basic science of neurodegenerative disease. As you understand the science, you can understand the targets, you can develop better therapies. In addition, these biomarkers that I mentioned a minute ago, it's now possible to measure in blood the rate of neuronal death. So think of HbA1c and diabetes. It's elevated a disease, a good drug could knock it down. The same thing with neurofilament light, or NFL, and neurodegenerative disease. It measures the rate of neuronal death. It's elevated a disease, a good drug could knock it down. That dramatically changes the economics, how rapidly you can get to a sense of whether your drug is working, the speed and the cost efficiency of development. And then one more recent trend is vectorizing antibodies in gene therapy. Antibody therapy requires an infusion 8 to 12 times a year. It's possible now, in the hot new area, growing area of gene therapy, to put the code of an antibody into a gene therapy vector, treat a patient once, and there's a therapeutic benefit for years. So those trends have initiated a tremendous amount of interest in pharma. This is going to be a rapidly growing space. And a lot of what I'll talk about in the next 25 minutes or so is how ProMIS is taking advantage of all of those trends to build a lot of value in the near term. Now, there's one other aspect of value, which, of course, economic value. So here's a map of the neurodegenerative space. The different diseases are in bubbles, and the bubble size corresponds to the value of that disease. The metric we've chosen to use is one that is relevant to what we're trying to do with our portfolio. Clinical proof of concept that de-risks a program enough to the point where there's a 60% chance or so of it being approved. The risk-adjusted NPV at that point is a very important number. In Alzheimer's, that risk-adjusted NPV is $12 billion, which is in line with what positive phase 2 results in the space have generated in the market. In Parkinson's, it's $3 billion. The other key to the neurodegenerative space in terms of value is there are no disease-modifying therapies, meaning there are no therapies that actually affect the root cause of disease. That's about to change, but let me get at what that root cause is. This is one of the most important advances in the science. All of these diseases that you see listed here, neurodegenerative and others, are caused by misfolded proteins. Those science pictures are an actual photo of the misfolded protein. Amyloid beta, tau, TDP-43, alpha-synuclein, some of these proteins will be familiar to you who've followed the space. As the phrase misfolded suggests, though, it also means there are forms of that protein that are not misfolded, that are critical for normal neuronal health. Some of these same proteins are responsible for synaptic remodeling, neuronal stability, DNA repair, RNA protection. You don't have to be a scientist to know that you don't want to interfere with those functions in your brain. So the critical insight about neurodegenerative disease is there are both pathological and biologically important aspects of these proteins. So what do we know about the foundation of neurodegenerative disease? The timing is excellent, the values are significant, and the science is clear. The key to neurodegenerative disease is to develop therapies that neutralize the misfolded toxic form, but stay away and don't interfere with the normal biologic form. And what ProMIS has is a unique capability and a very strong track record of creating multiple antibodies that do precisely that. That's the basis of our value creation. Now, in the remainder of my time, I'll focus more exclusively on ProMIS, and there are three key takeaway messages I want to leave with you this afternoon. One is that amyloid is a critical target in Alzheimer's disease. There's been a lot of controversy about that, but I'll show you the results of 43 in aggregate, 43 phase 2 and phase 3 trials that have dealt with amyloid, and those results are precisely in line with the science. There are successes and failures. The successes come when products are selectively targeted towards the misfolded form. Failures come when that's not the case. PMN310 is more selective than the programs that are looking successful, which is why we think we can be best in class. The second thing I'll focus on in a little bit less depth is every other area of neurodegenerative disease has similar science. It's likely to have a similar clinical history. In the amyloid field, there have been successes and failures. The same could be true in the alpha-synuclein field, TDP-43, and tau. To the extent that therapies are selective for the pathogenic misfolded form, they succeed. If not, they fail. And then the third key point I want to touch on just very briefly, but it's because it's become such a hot area in pharma. As you know, some of the hottest areas in pharma now are rare diseases, immuno-oncology, and gene therapy. Billions of dollars are pouring into it. A lot of big pharma companies like Novartis are making it sort of a core strategy. There's the opportunity to put antibodies into a gene therapy, which is a tremendous opportunity in neurodegenerative disease, but the criticality of selectivity is even greater there. In a gene therapy construct, if your antibody doesn't stay away from the physiologically important forms, there's even greater chance you're going to do harm. So our advantages are amplified in this hot area of gene therapy. Those are the 3 points I'll touch on. I'm going to go first into amyloid in our lead program, PMN310. First, let's talk about Alzheimer's. Now, this is also an opportunity for me to exemplify how we're going to use biomarkers to get very, very rapid value creation. Here's a picture of Alzheimer's, literally. Hopefully everybody in the room has a brain like the one on the left, control normal. 100 billion healthy neurons, that's what all that white space is. Over a 30-year period, the first half of which is asymptomatic, billions of neurons are killed. In the middle, that's mild cognitive impairment, first symptoms. And then on the far right, that's full-on Alzheimer's disease. The key to understanding Alzheimer's is something is killing billions of neurons over a 30-year period. What is that? It absolutely is started with toxic misfolded amyloid. There's a lot of evidence to that effect. Let me show you some of the most compelling evidence. There are two subpopulations, relatively rare subpopulations, whose contrast really illustrates the point. There was an Icelandic population that has a mutation so they never make amyloid. They were the inspiration for BACE inhibitors. No amyloid, never get Alzheimer's. In contrast, Down syndrome patients make excess amyloid, excess Alzheimer's. Down syndrome patients can have Alzheimer's in their teens. By age 40, everyone has pathologies. It can be asymptomatic. By age 55, 50% have symptomatic Alzheimer's. So the existence of amyloid predicts Alzheimer's disease, which is part of the evidence at its core. Now, what happens over these 30 years is amyloid misfolds, becomes a toxic oligomer, spreads around the brain, and it causes tau, which is usually inside the neuron, to become hyperphosphorylated and toxic as well. The two of them kill neurons, and that makes neuroinflammation go up. All three of those are important targets. We're working on all three of those, but amyloid is very much the primary target. Now, the reason why there's been any skepticism about that is people's interpretation of clinical trials. Well, let me show you the actual evidence from clinical trials. To the extent that clinical programs targeting amyloid are selective for the pathogenic misfolded form, they succeed. If not, they fail. So let's start with what are the three different types of amyloid? Monomer, which is produced every minute you're alive, that's physiologically important. Toxic oligomers, thousands of studies show that that's the bad actor. And plaque, which is a red herring. The most common strategy in the amyloid development program has been to target monomer. 26 phase 2 and 3 studies, 0 for 26. The second most common strategy has been to create an antibody that's not selective. It binds all forms of amyloid. 4 programs, 13 studies, 0 for 13. So going after the wrong form of amyloid or not being selective at all is 0 for 39 in the clinic. But there are two programs, aducanumab from Biogen and BAN2401 from Eisai, that are partially selective to the right side for oligomers and plaque. Their clinical results, 4 for 4. So the clinical results are right in line with the science. To the extent you get closer to being selective for what's killing neurons, the toxic oligomers, the therapy works. Now, Biogen made some pretty significant mistakes in their development program that require me to give you a little bit more explanation about why I call it 4 out of 4. Let's review the Biogen aducanumab program. This was the first ever successful study. It was the PRIME study, 166 subjects for Biogen. It came out in December 2014. It was the inspiration for ProMIS to do what we're doing. We were formed after this. Very, very strong dose response curve. Low is good. CDR sum of boxes, the endpoint there is what was used in their pivotal trial. And 10 mg was much better than 6, which was much better than 3. Based on that, Biogen decided to make an $800 million bet, 3,200 subjects in a pivotal trial to seek approval. But Biogen initially agreed to start that pivotal trial with 2/3 of the high-dose patients only getting 6 mg, not 10. They were worried about a side effect, ARIA-E. But obviously, 10 is much better than 6. That was compromising the trial. So halfway through, they corrected it. A V4 amendment, they called it, put all the high-dose patients on 10 mg. But then 18 months later, before any of those patients for V4 really had a chance to complete the trial, they conducted a pre-planned futility analysis based on the first half of the data and said the drug's not working. Well, it was only first half of the data using 6 mg data. They didn't wait for the 10 mg data to come in. Fortunately, during the 3 months we're doing that analysis in the first quarter of 2019, another 325 patients completed. It was enough high-dose 10 mg data that they knew that they had made a huge mistake. The drug actually works at the highest dose. And then what they showed in December is a new cut at the analysis where they only looked at patients who consented to the protocol, making all high-dose patients get 10 mg per kg. And both of those were successful studies. Aducanumab in 3 studies, if you adjust for Biogen's mistake, and it was a big mistake, but if you try to understand the question, does aducanumab work at 10 mg? 3 studies suggest yes, it provides a slowing of disease progression by about 25%, but with a side effect. The other thing that was true in the aducanumab pivotal is plaque binding didn't correlate with clinical response at all. More evidence that plaque is a red herring, plaque doesn't matter. If you had a version of aducanumab that didn't bind plaque and give more dose to the oligomer, you'd have a better drug, and that's exactly what we've created. So recapping quickly, this is a dot plot. A strong dot means a strong binding signal. So we're looking at the 4 different categories. Bapineuzumab is an example of the 0 for 13 non-selective category, binds all forms of amyloid. Solanezumab is an example of the 0 for 26 monomer-focused category. Aducanumab is an example of the 2 programs that are partially selective. They bind plaque and oligomers. We've created a product that only binds plaque. Now, the problem with binding plaque, we've created a product that only binds oligomers, the problem with binding plaque, as aducanumab and BAN does, is that you have a side effect that comes from that. You look at this vascular deposit binding on the left. These are slices of a brain of an Alzheimer's patient who died. That's the sort of thing that leads to ARIA-E. We have none of that. So we don't bind plaque. We probably won't create that side effect. Now let's talk about the oligomer. Remember those brains where you saw billions of neurons dying over a 30-year period? Look at the panel on the lower left. This is what oligomers, toxic oligomers of amyloid can do to neurons. On the left, you see a healthy neuron. Over 24 hours, that neuron's being killed, and on the right, you see a normal control. That's exactly what happens billions of times in Alzheimer's disease. Toxic oligomers. You can't do that with monomer. You can't do that with plaque. Toxic oligomers are the neuron killers. We also looked at how well our product binds to the actual toxic oligomers from human brains. High bar is good. We took a fraction of brain homogen of people who died of Alzheimer's that contained the toxic oligomers. We had a very strong binding signal, much stronger than aducanumab, which was much stronger than bapineuzumab. Now, PMN 310 was the best of over 300 antibodies we created looking for exactly this outcome. And in doing that, we were following the strategy that has led to the 3 biggest products in the history of the industry. Best in class, not first in class. Lipitor was the biggest statin 10 years after Mevacor. Humira was the biggest TNF-alpha antagonist 5 years after Remicade. In each case, the people who developed those products took a look at things that came first, said what makes them work and how could I make them better. They implemented that and they had a tremendous product. And that's exactly what we believe we've done in the Alzheimer's field. The second part of the equation, though, is how rapidly can you create value by showing that in the clinic? And that's where biomarkers are going to make a huge difference. This is an example of the type of readout we might see in our very first studies in the clinic with PMN 310, which could cost as little as $5 to $10 million. Multiple doses, showing a dose-dependent reduction in certain biomarkers. I'll pick one, neurofilament light. It's a direct measure that you can get in the blood of neuronal loss. It's elevated in disease. You could knock it down with a good drug. If you knock it down in a dose-dependent fashion, that's a very strong signal that the drug is doing what you want it to do. Going forward, then, not only will we capitalize on biomarkers, but also capitalize on another important development that can lead to speed in the Alzheimer's field, which is Alzheimer's and Down syndrome. So here's an overview of the 3-year plan. We need to do a little bit more work to get into the clinic. We'll first go to what's sporadic Alzheimer's disease, elderly patients with Alzheimer's, hopefully to prove that we have no safety issues, and hopefully to see some positive benefit on biomarkers. Then we'll go to Alzheimer's disease in Down syndrome. Remember I mentioned earlier, in Down syndrome, there's a very, very high prevalence of Alzheimer's disease. Rapidly progressive, everyone is in care. Some of the top Down syndrome Alzheimer's guys are on our advisory board. It's a community where you can show prevention because everyone is in care and everything's being monitored. It's also a disease subset of Alzheimer's where we believe the opportunity to move rapidly to approval is greatest, possibly with a label that says prevention. And once you have an approved product in Down syndrome Alzheimer's, you can expand that to sporadic Alzheimer's. So that's our game plan in Alzheimer's disease. Again, a quick summary of PMN 310. Failures of programs targeting the wrong form of amyloid, which are 0 for 39, investment of over $10 billion in the clinic, confirm the science. The toxic oligomer is the problem. Biogen aducanumab pivotal trial, despite some of the mistakes they made, is the largest study set in Alzheimer's disease. It looks approvable, and it confirms you can provide clinical benefit with symptomatic patients if you neutralize the toxic oligomer. We're highly selective for the toxic oligomer. We bind it better. That could give us the ability to be best in class, and we can demonstrate some clinical value in the very first studies for a relatively modest amount of capital. Now, more briefly, let me touch on one of our other programs that I think exemplifies what we're doing in the partnering area, and also it's an opportunity to talk a little bit about this trend of vectorization, alpha-synuclein. Misfolded alpha-synuclein is a critical problem in a number of diseases like Parkinson's and MSA. This is an example of some of the results. Remember, we've been talking about neuronal death. Look on the right panels. At the top is a healthy neuron that's in red. In the middle is what toxic oligomers of alpha-synuclein do, kill all the oligomers, no red. At the bottom, our antibodies can protect those neurons, dopaminergic neurons, from toxic oligomers. And what you see in the bars are 6 different antibodies. We now have 7 that do that in a statistically significant way. You need not only effectiveness, but you also need selectivity. Alpha-synuclein is a very complex challenge in terms of the need for selectivity. There's 6 forms. 2 of them, in red on the left, are physiologically important. You want to avoid binding them. 2 in the middle, in green, are the ones you want to bind, and you'd like to minimize binding to the ones on the right. We've been able to compare our antibody to competitive antibodies that have gotten partnering deals, and we have the best selectivity of all. All the antibodies, as you would expect, bind the toxic forms. We're the only ones who can avoid binding the plaque of alpha-synuclein, which is called Lewy bodies. And we're the only ones who fully avoid binding the physiologic form, although BioArctic, who partnered their program with AbbVie, do a pretty good job of that as well. So here's an example. It's true of all of our programs. We're able to create selectivity better than anyone else out there. Now, one of the first areas where someone was vectorizing an antibody was AbbVie in alpha-synuclein and the BioArctic deal. Let me show you a little bit about this trend. Over the last couple of years, there's been an explosion of interest. It started with AbbVie vectorizing a tau antibody. And again, what that is taking the DNA for that antibody that you could put into Chinese hamster ovary cells and manufacture it in a reactor. But you take that same DNA, put it in an adenoviral vector, you give it to a patient, one treatment, they can make the antibody on their own for years or possibly decades. AbbVie did that with Voyager in 2018. Novartis acquired Avexis, a gene therapy company, for $8.7 billion, partly for their SMA asset, but also to be able to vectorize antibodies. AbbVie did another deal in early 2019 with alpha-synuclein. And very recently, the FDA issued guidance about gene therapy because it's such a hot and growing area. They said in rare diseases, they encourage sponsors to have a first-in-human study that could be adequate for marketing approval. This is an area where you can have what's called a one-and-done clinical program. Now, what we've seen in our partnering discussions is there was some interest at JP Morgan 2019. We were raising our differentiation. 2020, going back to JP Morgan, every single partner we talked with is interested in gene therapy. For all but one, it was their primary strategy. So this is really taking off as an area of interest for pharma. And here's an example of a development plan we talked about with a couple of those partners. In alpha-synuclein, it combines a few of the factors I've talked about. Multiple system atrophy is a very aggressive, rare disease driven by alpha-synuclein. Time from diagnosis to death is about 6 to 8 years. Biomarkers are elevated. So a game plan could be go into the clinic with an antibody, hopefully show some benefit without any adverse events. In parallel, develop a gene therapy program. If things look good, switch to gene therapy and possibly have a one-and-done trial. Very rapid route to approval, de-risked early on, very attractive to large pharma because they see the benefit of not having to have managed care pay for 8 to 12 infusions a year, one shot, and a patient's good for many, many years. But the core point in terms of vectorizing for gene therapy is in a gene therapy vector, drug is going to get inside the neuron where a lot of these physiologic roles happen. If you don't select away from, if you impair in any way the important biologic form of these proteins, you can cause serious adverse events. So in areas like amyloid beta, TDP-43, alpha-synuclein tau, if you want to vectorize, you need antibodies as selective as what we can create, and we have tremendous advantages. So the advantages we have in antibody therapy are magnified as pharma moves more and more towards gene therapy strategies. Let me summarize with a picture of catalysts and then come back to those 3 key takeaways. For the last couple of years, to be totally frank, some of you have been following the story for a while, mistakes and miscommunications by both Biogen and Eisai have really dampened capital availability in the Alzheimer's field generally and amyloid in particular. I think we're moving into a new era. We see very likely Biogen will file for a BLA for aducanumab by the end of this month or soon thereafter. That'll be kind of something that puts exposure on. If you do amyloid right, it can work. They may have an advisory committee. We expect, and so do many others, an aducanumab approval in the beginning of 2021, which will be a tremendous catalyst for the field generally and for us. So as we generate our clinical data, it'll be against the back of an initial approval which has some liabilities, a side effect and efficacy not where we'd like it to be. And I've already shown you we have scientific evidence suggesting we could be much, much better in the clinic. So we could see some tremendous catalysts coming out of that data. In parallel with that, we have very active partnering discussions going on, alpha-synuclein, TDP-43, and tau. In every case, companies are interested in gene therapy. They're trying to figure out how that fits the overall equation, but we're differentiated. We continue to expand our portfolio. That could lead to more partnering options, and as we get better capital formation, we'll probably take more than one antibody forward into the clinic. So again, back to the 3 key points, and then I'll conclude with this. Amyloid is a very, very important target, the primary target in Alzheimer's, but you have to do it right to be successful. 43 clinical trials and all the science tell us the same thing. We think we've done it more right than anyone else. We've created an antibody, PMN310, that's highly selective only for the toxic oligomer and does a very, very good job binding that. And over the next 2 or 3 years, we think there'll be a tremendous amount of data, both external and internal, that sort of shows the value of that. But it's also important to mention that the amyloid clinical experience, which has had a lot of frustration and failure in addition to successes, is probably going to play out in these other categories. The science is the same. The degree of selectivity for the toxic misfolded form correlates with the degree of clinical success. And particularly that's going to be true as gene therapy, particularly vectorizing antibodies, takes off. It's even more important to be highly selective in that scenario, which has tremendous potential benefits, avoiding infusion costs, maybe better efficacy, but puts the even higher critical need on making sure that you have a ProMIS level capability in terms of being able to create highly selective antibodies. So thanks for your attention. We're looking forward to the next few months and years. I think it's going to be a great time in the neurodegenerative space and a great time for ProMIS. I really appreciate your attention here at Noble. Go gamble. Thanks. Appreciate it. I think we're probably out of time. So if anybody has any questions, I'd love to sort of catch you in the hall. But again, thanks very much. Appreciate it.